This video will cover subtopic 8.2, cell respiration, and we'll go through the steps involved with glycolysis, the Krebs cycle, and the electron transport chain. So it's a bit of a long video, so buckle in. So, list of understandings that we'll cover in this video, as well as applications and skills. So we'll start with a bit of information about ATP and phosphorylation, just a few things to recall. So just remember that adenosine triphosphate, ATP, is a high-energy molecule that functions as a power source for cells. One molecule of ATP contains 3 covalently bonded phosphate groups, which stores potential energy in their bonds. Phosphorylation makes molecules less stable, so this process of adding a phosphate to a molecule will make it less stable, which means that ATP is a very reactive molecule that contains high energy bonds. When ATP is hydrolyzed to form adenosine diphosphate and an inorganic phosphate, the energy stored in that terminal phosphate bond is released for use by the cell. So this diagram just outlines where those phosphates are in ATP. So here we have adenosine triphosphate, here we have our adenine molecule, it's connected to a ribose, and that ribose is covalently bonded to those phosphate molecules. So the more phosphates you tack on, the more energy that's stored in between those covalent bonds between the phosphate groups. Now, the highest energy is found between the second and third phosphate group in that bond. When that bond gets cleaved, you produce adenosine diphosphate and an inorganic phosphate. That energy that was contained within those bonds gets released, and that energy is what will act on other molecules. So in this cycle of ATP that gets cleaved to produce ADP and inorganic phosphate, these two molecules will then be used again to generate new ATP, which will be used to generate the work or the energy needed for cellular work. So with the functions of ATP, the energy currency of the cell by releasing energy when hydrolyzed to ATP is what powers cell metabolism. This may transfer, so ATP may transfer the released phosphate group to other organic molecules, rendering them less stable and more reactive. Remember, that process of phosphorylation makes molecules less stable. ATP is synthesized from ADP using energy derived from one of two sources. So that energy source can be solar energy, and photosynthesis will actually convert light energy into chemical energy that is stored as ATP. This is the first part of photosynthesis, the light-dependent reactions that will generate ATP. And then there's the oxidative process where cell respiration breaks down organic molecules to release chemical energy that is stored as ATP. So this oxidative process that uses oxygen and redox, a series of redox reactions, is what we will go through in this video for generating ATP. So in this process of cell respiration, this is the controlled release of energy from organic compounds, and we'll use glucose in our example here to produce ATP. So recall that there are two forms of cell respiration. There's anaerobic respiration and aerobic respiration. So anaerobic respiration involves the incomplete breakdown of organic molecules for a small yield of ATP. In anaerobic respiration, there is no oxygen required. And in this form of cell respiration, only a small amount of ATP is generated. We'll have a closer look at that later on in the video. Aerobic respiration involves the complete breakdown of organic molecules for a larger yield of ATP. This is where oxygen is required. In cell respiration, the breakdown of organic molecules will occur through a number of linked processes that involve a number of discrete steps. So there's 3 parts to cell respiration, and each of them involve a number of small steps. By staggering the breakdown, the energy requirements are reduced. The activation can be divided across several steps. The released energy is not lost through this staggered breakdown. It is actually transferred to activated carrier molecules through oxidation reduction reactions, or what are commonly called redox reactions. So in this diagram here, what it shows is that if we started with our sugar and oxygen, and just were to directly release that energy stored within that glucose, there'd be a large amount of energy released. So here we have our reactants and our products. Our products are CO2 and H2O, which are at a lower energy level than sugar, so it contains less energy. So this energy release would be pretty uncontrolled, and a lot of it would be lost. Here we have a staggered breakdown of our reactants. So each of these smaller steps here represents a discrete step in releasing that energy. And these smaller steps allow a more controlled release of energy and allow that energy to be more efficiently harnessed in creating ATP. So this is why it's not just one step of breaking down that glucose. It occurs over multiple steps through multiple processes. So it is redox reactions that will allow this energy to be harnessed. So when organic molecules are broken down by cell respiration, the chemical energy is transferred by means of redox reactions. Redox reactions involve the reduction of one chemical species and the oxidation of another. So redox reactions happen in conjunction with each other. So if you have oxidation, you also have to have reduction. So most redox reactions typically involve the transfer of electrons, hydrogen, or oxygen. So what is a reduction reaction? What is an oxidation reaction? So a reduction reaction is typically the gain of electrons. So if something is being reduced, it is gaining electrons or hydrogen, or reduction is referred to as the loss of oxygen. So it can be any one of those. So reduction is a gain of electrons, a gain of hydrogens, or a loss of oxygen. Oxidation is the loss of electrons. So if something is being oxidized, it is losing electrons. Oxidation is also the loss of hydrogen ions or the gain of oxygen atoms. And so this table here just compares oxidation and reduction reactions. So here, if oxidation, so something is losing electrons, there's going to be something that gains those electrons. So those electrons, when they're lost, they don't just kind of go out into space and disappear. Those electrons get transferred over to another chemical or another molecule. So if that thing is losing electrons, something else is going to be gaining electrons. If it's oxygen that's involved, so in oxidation, it's a gain of oxygen atoms, and those oxygen atoms don't have to come from somewhere. So they come from the molecule that is being reduced. So that molecule will lose electrons. Same thing with hydrogens as well. So there are ways to remember oxidation and reduction, which you'd have to do for this for cell respiration, but you also have to do it in electrochem as well. So there is that mnemonic oil rig, which means oxidation is loss, reduction is gain. So if you remember oil rig, you can remember that oxidation is a loss of electrons and reduction is a gain of electrons. Now, if you're offended by fossil fuels and the idea of oil deeply insults you, then you can remember Leo the lion goes grr, or Leo goes grr. So Leo, loss of electrons is oxidation, and grr, gain of electrons is reduction. So Leo the lion says grr. And if you find lions scary, well, then you can remember the phrase Elmo. So electron loss means oxidation. So with these redox reactions now, they occur involving what are called hydrogen carriers. So cell respiration will break down organic molecules and transfer hydrogen atoms and electrons to carrier molecules. So this is the redox part here. So as that glucose is broken down, hydrogens are removed from it, electrons are removed from it, and transferred to a different molecule. As the organic molecule, for example glucose, is losing hydrogen atoms and electrons, this is the oxidation reaction. Energy stored in the organic molecule, in this case glucose, is transferred with the protons, hydrogen ions, and electrons to carrier molecules. And we'll talk about why they're called carrier molecules in a second. Now, the carrier molecules are called hydrogen carriers or electron carriers as they gain electrons and protons, those hydrogen ions. So the most common hydrogen carriers is a molecule referred to as NAD+, which is reduced to form NADH. So there's two forms of this molecule. There's the NAD plus, and then the reduced form, the NADH, when it gains an electron and a hydrogen. Now there is another carrier, which is less common, called FAD. And FAD gets reduced to form FADH2. So this is the reduced form when it's gained electrons and hydrogen ions, and this is the oxidized form. So what are these carriers? These hydrogen carriers, they kind of function like taxis in a way. They will transport electrons and the hydrogen ions to the mitochondria. So this process of glycolysis is occurring out in the cytoplasm as those hydrogen ions and electrons get removed from glucose and transferred over to NAD or FAD, in this case we'll say NAD. NAD will then transport or carry those hydrogen ions and electrons to the mitochondria. And specifically, they will carry them to the cristae of the mitochondria. Now, the cristae of the mitochondria is the site of the electron transport chain, which we'll get into a little bit later. And this will use the energy transferred by the carriers to synthesize ATP. So these hydrogen ions and electrons will be used to create that ATP, and we'll go through how that's done specifically as well. So this process requires oxygen to function, and hence only aerobic respiration can generate ATP from hydrogen carriers. This is why aerobic respiration unlocks more of the energy stored in the organic molecules and produces more ATP. So this diagram here just shows the oxidation reduction reactions that are occurring involving the organic molecule, glucose, and NAD. So here we have the hydrogen ions here and the electrons that get transferred from the organic molecule to NAD. And this is, the NAD is undergoing the reduction reaction. So this is what's showing here. So here, this is its oxidized form. And then here, it's gained that hydrogen ion and electron, and is now reduced. The organic molecule undergoes oxidation as it is losing the electron and hydrogen ion and has had those transferred over to NAD. So this can lead us now into talking about the first step in cell respiration, which is glycolysis. So the main compound used in cell respiration is glucose, although lipids and proteins can be used, but your cells would prefer glucose. Lipids are not preferentially used as they are harder to transport and digest and break down. So they will act as a long-term energy storage, but it's harder for your body to use them. Proteins are not preferentially used as they release potentially toxic nitrogenous compounds when broken down. So they'll release those amino groups found within those amino acids. So the first step in the controlled breakdown of carbohydrates is glycolysis. This is the first processes which occurs in the cytosol of the cell, and this part uses no oxygen. So this part can happen in the presence of oxygen or in the absence of oxygen. So in glycolysis, you have a hexose sugar, a 6-carbon sugar, which is going to be broken down into 2 molecules of pyruvate, a 3-carbon compound. So basically, you're taking this chain of 6 carbons, and then you're going to break it down into a chain, 2 3-carbon chains. So in glycolysis, two molecules of ATP are used to begin the process. So why are you using ATP? If the whole process is to generate ATP, you might ask. So the analysis there, or the analogy, sorry, there is that, with money, for example, you gotta spend a little to make a lot. So this is kind of an investment of energy to gain a lot of energy in the end. So we're spending a little to get a lot. So in the first reaction of glycolysis, the phosphates from ATP are added to glucose to form a molecule called fructose 1,6-bisphosphate. So here we have our 6-carbon glucose. 2 ATP are used. They generate 2 ADP, and we add on 2 phosphates, and the 2 phosphates get added on to the 1 carbon and the 6 carbon. So we get fructose 1,6-bisphosphate. So the importance of phosphorylation, and phosphorylation is that we're adding on these phosphate groups, and the importance of phosphorylation in this step is that it creates a less stable molecule. So we've increased the energy, which makes it less stable. The less stable 6-carbon phosphorylated fructose is then split into 2 3-carbon sugars called glyceraldehyde 3-phosphate. So through an enzymatic reaction, this 6-carbon compound gets cleaved to form 2 molecules here of glyceraldehyde 3-phosphate. And this is actually a simplified version of this. So it's not quite that direct. You generate kind of an intermediate molecule in one of these steps, which is not glyceraldehyde 3-phosphate, and then there's another step to convert that intermediate into glyceraldehyde 3-phosphate. But the end result of this is that you have 2 glyceraldehyde 3-phosphates, also referred to as G3P. Now, each glyceraldehyde 3-phosphate will then undergo oxidation to form a reduced molecule of NADH. So this is our first instance here of NAD undergoing reduction to generate NADH. So we have electrons and hydrogens coming off of glyceraldehyde 3-phosphate that will reduce NAD to NADH. As NADH is being formed, released energy is used to add an inorganic phosphate to the remaining 3-carbon compound. So we take an inorganic phosphate and tack it on to that 3-carbon compound. So we'll have 2 phosphates on there. Enzymes will then remove the phosphate that has been added on and add it to ADP to produce ATP. This is referred to as substrate level phosphorylation, and we'll have a look at this in a diagram. The end result is the formation of 4 molecules of ATP, 2 molecules of NADH, and 2 molecules of pyruvate. Once pyruvate is obtained, the next pathway is determined by the presence of oxygen. If oxygen is present, pyruvate will then enter the mitochondria and aerobic respiration occurs. If oxygen is not present, anaerobic respiration occurs in the cytoplasm. So the diagram here for this particular step of generating pyruvate, and pyruvate is kind of the molecule that we need to initiate the next step, is we have our glyceraldehyde 3-phosphate. We have 2 of them. And here we have our NADH coming along, taking off an electron and a hydrogen. And it does this. We have 2 NAD molecules that do this because we have 2 glyceraldehyde 3-phosphates. And at the same time, the energy in this process allows 2 inorganic phosphates to be tacked on to each glyceraldehyde 3-phosphate. So now each glyceraldehyde 3-phosphate will now have 2 phosphates attached to it. This will then, through enzymatic reactions, will allow ADP to obtain those phosphates and become ATP. And in the process, this will generate 2 pyruvates. So at this stage here, we can talk about the continuation of aerobic respiration, and this is where anaerobic respiration would start if oxygen was not present. So in anaerobic respiration, if oxygen is not present, pyruvate is not broken down further and no more ATP is produced. The pyruvate remains in the cytoplasm and is converted into lactic acid, or ethanol and carbon dioxide, in plants and yeast. This conversion is reversible and necessary to ensure that glycolysis can continue to produce small quantities of ATP. So here is our process of glycolysis. Here we're generating pyruvate, and here, we don't have any oxygen to allow pyruvate to continue on. So in the absence of oxygen in animals, that pyruvate will then undergo more oxidation and reduction to generate lactate. This is a 3-carbon molecule, that lactic acid. And in plants and yeast, that pyruvate will undergo fermentation or further reduction through the loss of a carbon dioxide to generate ethanol. And this will allow a small amount of NAD to be regenerated, and will allow glucose to continue to be broken down. In anaerobic respiration, glycolysis involves the oxidation reactions that cause NAD to be reduced to NADH. NADH is oxidized via aerobic respiration to restore available stocks of NAD. In the absence of oxygen, glycolysis will quickly deplete available stocks of NAD, preventing further glycolysis. Fermentation of pyruvate involves a reduction reaction that oxidizes NADH to NAD+. This aerobic respiration allows small amounts of ATP to be produced in the absence of oxygen, so that ATP that would be produced through that first step in the breakdown of glycolysis will continue to be produced. So looking at a summary of glycolysis, we have 2 ATPs that are used to start the process. A total of 4 ATPs are produced, resulting in a net gain of 2 ATPs, so we've doubled our investment. 2 molecules of NADH are produced. Just recall that the pathway here for this first process here of glycolysis, this pathway occurs in the cytoplasm of the cells. And this metabolic pathway is controlled by enzymes. So involved in all of these steps are enzymes, which are not mentioned, but they are there. The 2 pyruvate molecules are present, that are generated, are present at the end of the pathway. The generation of pyruvate ends that process of glycolysis. In the next step of cell respiration is the link reaction. This is the first stage of aerobic respiration. And that first stage of aerobic respiration is referred to as the link reaction, which transports pyruvate into the mitochondria. So in the matrix of the mitochondria, pyruvate is decarboxylated to form the 2-carbon acetyl group. The removed carbon is released as carbon dioxide. The acetyl group is then oxidized with the formation of reduced NAD+. Finally, the acetyl group combines with coenzyme A, abbreviated as CoA, to form acetyl-CoA. As glycolysis splits glucose into 2 pyruvate molecules, the link reaction occurs twice per molecule of glucose. Acetyl-CoA can enter the Krebs cycle to continue aerobic respiration, or can be synthesized into lipid for storage purposes. So if ATP levels are high in the cell, then at this stage here, the acetyl coenzyme A can be used for lipid storage, or converted into lipid for energy storage. So in the link reaction here, we have our pyruvate molecule. So we have our 3-carbon compound that's currently in the cytosol. As this gets transported into the mitochondria, a series of reactions will occur in which NAD will get reduced. Pyruvate will lose a carbon to generate CO2, and then we'll add on this rather big molecule called coenzyme A onto this to generate acetyl coenzyme A. So here is our 2-carbon compound. This is the acetyl group, and then here we have coenzyme A. So once acetyl-CoA has been generated, this can then enter into the next stage of cell respiration, which is referred to as the Krebs cycle. So the second stage of aerobic respiration is the Krebs cycle, which occurs within the matrix of the mitochondria. It's also referred to as the citric acid cycle, or the tricarboxylic acid, the TCA cycle. So there's a few names for it. It's all the same thing. So in the Krebs cycle, acetyl coenzyme A transfers its acetyl group to a 4-carbon compound, referred to as oxaloacetate, to make a 6-carbon compound called citrate. So the starting compound here is this 4-carbon compound called oxaloacetate. And this will combine with the 2-carbon compound, acetyl-CoA, to generate a 6-carbon compound. In the process, coenzyme A is removed, and we have a 6-carbon compound called citrate. Citrate, this 6-carbon compound, is then oxidized to form a 5-carbon compound. The carbon is released as carbon dioxide after combining with oxygen. As the 6-carbon compound is oxidized, NAD+, another NAD+, is reduced to NADH. So this is kind of the controlled energy release that is going on here in the breakdown of this carbon compound. So here we've generated our citrate, that 6-carbon compound. Now this will get oxidized, and as it gets oxidized, a carbon is lost in the form of CO2. So we go from a 6-carbon compound to a 5-carbon compound. And as it gets oxidized, this is where NAD plus gets reduced to NADH. So we have another transfer of electrons and hydrogen. So we have another bit of energy that's transferred. The 5-carbon compound is then oxidized and decarboxylated to form a 4-carbon compound, and another NADH is reduced to form another NAD plus is reduced to form NADH. The removed carbon combines with oxygen and is released with carbon dioxide. So we have our 5-carbon compound here. Another carbon is removed or released in the form of carbon dioxide, creating a 4-carbon compound. And as that 4-carbon compound gets created, NADH plus, or NAD plus, is reduced to NADH. So another set of electrons, or another electron and hydrogen, is transferred to NADH in this process. So another bit of energy is released. Now at this point we have a 4-carbon compound. Remember, our starting product here was a 4-carbon compound. So the rest of this cycle, and we'll probably get a bit more energy out of this, but the rest of this cycle is taking this 4-carbon compound and converting it back into our starting 4-carbon compound, oxaloacetate. So at this stage, we don't have oxaloacetate, but we got to convert this back into oxaloacetate. So the 4-carbon compound undergoes various changes, resulting in several products. And as this occurs, another NADH is produced. The coenzyme, the other hydrogen carrier, FAD, is reduced to form FADH2. ADP is reduced to form ATP. So we generate another ATP, another substrate level phosphorylation from this, in going back to oxaloacetate. The 4-carbon compound is changed during these steps to reform the starting compound of the cycle, oxaloacetate, hence a cycle, which may then begin the cycle again. So as we regenerate that oxaloacetate, this is available to bind with the next acetyl-CoA to start this whole cycle all over again. So in this process, we've generated a few NADHs, we've generated an ATP, and we've generated an FADH2. So remember that the Krebs cycle will run twice for each glucose molecule entering cell respiration. Remember, we generated 2 pyruvate molecules. Each pyruvate will cause 1 turn of the Krebs cycle. So each glucose molecule will cause 2 turns of the Krebs cycle. So the following products results from the breakdown of one glucose molecule. So we've generated 6 molecules of ATP so far for a net gain of 4 ATP. We have generated 6 molecules of NADH and 2 molecules of FADH2. We've also generated 6 molecules of carbon dioxide, 2 from the link reaction and 4 from the Krebs cycle. So far, only 4 ATP have been produced through substrate-level phosphorylation. The breakdown of each glucose molecule results in a net gain of 36 ATPs. So the majority of the ATP produced by is going to be produced by oxidative phosphorylation, part of the final phase of cellular respiration. So all of these NADHs and these 2 FADH2s are going to be used in this final step of cell respiration to help create large quantities of ATPs. And this final step in cell respiration is referred to as the electron transport chain. So this is the final stage of aerobic respiration, which is located in the inner mitochondrial membrane. So this is within the cristae. So the inner membrane is arranged into folds, those cristae, which will increase the surface area available for the electron transport chain. So by having all those folds, you can fit a lot more of these proteins involved in that electron transport chain, kind of maximizing how much you have going on. The electron transport chain releases the energy stored within the reduced hydrogen carriers in order to synthesize ATP. Basically, what it's saying here is that those hydrogens and electrons within those carrier molecules get released, and as they are released, this is going to help synthesize ATP. This process is called oxidative phosphorylation, as the energy to synthesize ATP is derived from the oxidation of the hydrogen carriers. And this will occur over a number of steps, which shouldn't come as a surprise at this point. As I mentioned, that whole process of cell respiration is a series of small steps. So this electron transport chain is going to be a series of small steps. So embedded in the membrane of the mitochondria are molecules that are easily reduced and oxidized. These carriers of electrons are close together and pass the electron transport chain from one to the other because of an energy gradient. Each carrier molecule within that membrane has a slightly different electronegativity. So electronegativity refers to the ability to attract electrons, and because each molecule has a slightly different electronegativity, each molecule therefore has a different attraction for electrons. Electrons pass from one carrier to another because the receiving molecule has a higher electronegativity and thus a greater attraction for electrons. In the process of electron transport, small amounts of energy are released. So this diagram just kind of shows that energy gradient involved in the electron transport chain. Here are the different proteins involved in that electron transport chain. And each of these have a different electronegativity, so a slightly higher electronegativity than the previous protein or the previous molecule. So the electrons that get dropped off here at the beginning are going to move from one protein molecule to the next because this guy has a greater attraction, and this guy has a greater attraction for the electrons, so the electrons will move from this guy to this guy, and so forth. And as these electrons move from each protein, there is a small amount of energy that gets released. And so as that small amount of energy gets released, that will then be used. So oxidative phosphorylation occurs over a number of distinct steps. So step 1 is referred to as generating a proton motive force. So using those protons, those hydrogen ions, to create a motive force. So the hydrogen carriers, the NADH and FADH2, are oxidized and release high-energy electrons and protons. So here's our NADH. It will be oxidized to produce NAD. So as it's oxidized, it's releasing its hydrogen ions and its electrons. Now the electrons are transferred to the electron transport chain, which consists of several transmembrane carrier proteins. So here the electron is released and transferred to that first protein in that electron transport chain. So as the electrons pass through the chain, they lose energy. So the electrons will start here. And remember, this guy has a higher affinity for electrons. So the electrons will pass from this guy here to this protein. And as the electrons pass from one protein to the other, or from one carrier protein to the other, energy is lost. Now, as that energy is lost by the electrons, that energy is actually used to take the hydrogen ions that have been dropped off, and they will transfer or pass or transport, those hydrogen ions from one side of the mitochondrial membrane to the other side. And this creates a high concentration of hydrogen ions on one side of the membrane and keeps a low concentration of hydrogen ions on the other side of the membrane. This is a form of active transport where hydrogen ions are being transported against its gradient from a low concentration to a high concentration. And remember, in that transport from low concentration to high concentration, this requires energy to do this. Now, in the previous parts of the course, we would have talked about the fact that it would be ATP that would be used to drive this, but because we're going to be generating ATP, we don't want to be using ATP at this stage. So we can harness the energy within those electrons to drive those hydrogen ions from one side of the membrane to the other side of the membrane, creating this high concentration of hydrogen ions. Now, the accumulation of those hydrogen ions, those protons, within the intermembrane space creates an electrical gradient, electrochemical gradient, or a proton motive force. So we have this accumulation of these hydrogen ions here, and they're going to, as they have a high concentration there, and they want to move away from each other. The natural tendency is to kind of diffuse or spread out. The mitochondria will harness this energy here that's created in this proton motive force to now create ATP. So step 2 in this electron transport chain is ATP's synthesis through this process of chemoosmosis or chemoosmosis. So the proton motive force will cause hydrogen ions to move down their electrochemical gradient and diffuse back into the matrix. The diffusion of protons is called chemiosmosis, as it is facilitated by the transmembrane enzyme ATP synthase. So this enzyme here, this ATP synthase enzyme here, will allow these hydrogen ions to move from a high concentration to a low concentration, which is what they naturally want to do. Now, because this is an energy releasing process, as the hydrogen ions move through the ATP synthase, they trigger the molecular rotation of the enzyme synthesizing ATP. So as these hydrogen ions move through the ATP synthase, it will literally cause a rotation, a turning of this molecule, and this molecule, or this enzyme, will take ADP and an inorganic phosphate, jam them together to create ATP. So it will harness that energy of those protons moving from high concentration to low concentration to take ADP and bond it to an inorganic phosphate, creating ATP. So this is another look at the ATP synthase. here. So, there's a lot of different components to it, but basically you have your intermembrane space here of the mitochondria. You have the mitochondrial matrix here. So we have our high concentration of hydrogen ions. And as they move through this ATP synthase, this will cause rotation, which will take that ADP, an inorganic phosphate, and create ATP. So the third step of the electron transport chain is the reduction of oxygen. So if you'll remember now, we've had these electrons, or this electron, moving through our electron transport chain, and it will eventually reach that final carrier protein, and the electron will basically sit there until it has some place to move to. So in order for the electron transport chain to continue functioning, the de-energized electrons have to be removed. So that final electron has to be removed in order for this chain to continue moving. Oxygen acts as the final electron acceptor, so that electron will move from that membrane protein to oxygen, removing the de-energized electrons to prevent the chain from becoming blocked. Oxygen also binds with free protons in the matrix to form water. So those protons that are moving through that ATP synthase, as they move across that membrane, they will combine with oxygen and the electrons to form water. So doing so will remove those matrix protons, maintaining the hydrogen gradient. Remember, the idea here is to keep hydrogen ion concentration low here, allowing it to be high on the other side of the membrane. In the absence of oxygen, hydrogen carriers cannot transfer energized electrons to the chain, and ATP production is halted. Basically, in the absence of oxygen, you don't have an oxygen here, you don't have that final electron acceptor. So eventually this chain will become blocked, and you can't generate high levels of ATP. So this diagram here provides an overview of the electron transport chain. So here we have our mitochondrial matrix, we have our intermembrane space, here we have our inner mitochondrial membrane, here are our various carrier proteins. Here we have NADH coming in to the mitochondria and dropping off its electrons. The electrons were represented by these black arrows here, and hydrogen ions. As those electrons move from one carrier protein to the next, this will allow the hydrogen ion that has been dropped off to be transported to that intermembrane space, allowing or creating a concentration gradient, or that proton motive force, where you have a high number of hydrogen ions in this intermembrane space. Now, this process continues down that electron transport chain, allowing more hydrogen ions to move through that inner mitochondrial membrane. Now here we have our ATP synthase that will allow those hydrogen ions to move from high concentration to low concentration. And as they move, that energy that is released will help cause rotation of that ATP synthase, generating ATP from ADP and that inorganic phosphate. In that final carrier protein in the transport chain, this is where the electron will then be transferred to oxygen, and any hydrogens in the mitochondrial matrix to generate water. So this diagram here just provides an overview of where cell respiration occurs. So here we have our cell membrane. Here is the cytosol, or the cytoplasm of the cell. And then here we have the mitochondria. So here's the mitochondrial membrane. And then here is the inner mitochondrial membrane. So here's the cristae. That's all folded, hence why it looks folded. So in the cytosol is where we have glycolysis occurring. So this is where glucose will enter into glycolysis to generate pyruvate. That pyruvate is then transported into the mitochondria, where it undergoes that link reaction. To generate acetyl coenzyme A, and then acetyl coenzyme A can then enter into that citric acid cycle, that Krebs cycle, generating more NADs and FADs. And the NADs, the NADHs that are generated from glycolysis will transfer or transport those hydrogens and electrons from the cytosol into the mitochondria and into the mitochondrial matrix. And then here is where we get the electron transport chain on that inner mitochondrial membrane. And it's in the intermembrane space where we generate our hydrogen ion concentration, so that proton motive force that will ultimately be used to create ATP. So this chart here just provides a summary of the ATP production in cellular respiration. So, this part here looks at the energy flow in the general process. So we have energy contained within glucose, which then gets transferred to NADH and FADH2, which then gets transferred into that electron transport chain, which is then used for chemiosmosis, which then gets transferred to ATP. So here is our general energy flow. And then this chart here looks at ATP production in cellular respiration. So in glycolysis, we have 2 ATP produced, 2 ATP that's used, 4 ATP that gets produced, for a net gain of 2 ATP. In the Krebs cycle, we don't have any ATP that's used. We generate 2 ATPs, and we have a net gain of 2. And then in the electron transport chain and through chemiosmosis, this is where we generate the majority of our ATPs. So we'll generate about 32 ATPs, which will give us a total net gain of 36 ATPs. In total, a glucose molecule will produce about 38 ATPs, but because we've used 2 to start the whole process, we get a net gain of 36 ATPs. So, that's not a bad investment, not a bad return on investment, as we invested 2 ATPs and we got 38 out of it for a net gain of 36. So, from the table, there is a net gain of 36 ATP from cellular respiration. In reality, the number is closer to 30. This is probably due to some hydrogen ions moving back into the matrix without going through the ATP synthase channel. The 30 ATPs generated by cellular respiration account for approximately 30% of the energy present in the chemical bonds of glucose. The remainder of the energy is lost from the cell as heat. So even though it's kind of a controlled series of reactions, only about 30% of that energy is able to be harnessed. So, it's still a rather inefficient process, but it's the best that can be done, I guess, and enough to allow the cell to function properly. So one of the things you'd be expected to do if you were in HL Bio and writing that exam would be to analyze diagrams of the pathways of aerobic respiration to figure out where decarboxylation and oxidation reactions occur. So remember that with decarboxylation, carbon atoms are removed from the glucose molecule to form CO2. In oxidation, electrons and hydrogens are removed from glucose and taken up by NADH and FADH2, or taken up to produce these molecules here. And phosphorylation is the energy released from the breakdown of glucose and is used to phosphorylate ADP to make ATP. So this diagram here is just a summary of the stages of aerobic respiration, just showing where everything gets generated from. So we have glucose entering into glycolysis, which generate a net 2 ATPs, which also generate 2 NADHs. That glycolysis results in the generation of two pyruvate molecules, which go into the link reaction, which will generate two NADHs, but will also generate two CO2s. At the end of that link reaction, we've generated two acetyl coenzyme A molecules, which then enter into the Krebs cycle. From the Krebs cycle, we generate two ATPs as well as four CO2s. We also generate six NADHs and two FADH2. Now, those carrier molecules all drop off those hydrogen ions and electrons in the electron transport chain, which require an input of six oxygen molecules. And out of the electron transport chain, we get 32 ATPs and 6 H2Os. So if you look at that overall equation for cell respiration, so we have that glucose molecule, that C6H12O6, generating 6 CO2. So if we add together those CO2s, we get 6 CO2s plus 6 H2Os here. So those are where the different bits come from in terms of those products. Lastly, we'll have a look at mitochondria. So just recall that mitochondria are the power plants of the cell. The whole thing we've been talking about here is generation of energy. And it will function to synthesize large amounts of ATP through aerobic respiration. All eukaryotic cells possess mitochondria. Mitochondria are thought to have once been independent prokaryotes that were internalized by eukaryotes through that process of endosymbiosis. And the evidence for this is that they have a double membrane structure and they have their own DNA. It's circular and naked, and their ribosomes are 70S ribosomes as opposed to those eukaryote ribosomes, which are 80S. Their metabolic processes are susceptible to certain antibiotics, suggesting they were once prokaryotes. So the structure of the mitochondria is actually adapted to perform its function. So the outer membrane will contain transport proteins that enable the shuttling of pyruvate from the cytoplasm. The inner membrane contains the electron transport chain and ATP synthase. And the cristae, now the inner membrane is arranged into folds, cristae, that increase the surface to volume ratio. So it helps increase that surface area. The intermembrane space is a small space that maximizes the hydrogen ion gradient that gets created. And the matrix now, the central activity that contains the appropriate enzymes and the suitable pH for the Krebs cycle to occur. So this diagram here just looks at the various structures of the mitochondria. So here we have that outer membrane that contains those transport proteins for shuttling pyruvate into the mitochondria. Here we have the inner membrane that contains the electron transport chain and ATP synthase. Now that inner membrane is contains a lot of folds, or is highly folded, so that cristae, so that increases the surface area. And then the intermembrane space is going to be small, so it can quickly accumulate protons and create that high proton concentration gradient. And then here we have the matrix, so this is where like the Krebs cycle occurs, and this will contain the appropriate enzymes and a suitable pH to allow for the Krebs cycle to occur. So one of the things that you would be expected to do in HL Bio is be able to annotate a diagram to indicate the adaptations of mitochondria to its functions. So, if you want more information on this, have a look at page 387. And this also relates to the nature of science statement. So, this now ends the video for 8.2, dealing with cell respiration. I'd also like to take a second to thank BioNinja for providing a lot of diagrams and a lot of the information that was used in this video.